introduce our speaker. Ashish.
Thank you, Ileana. Professor Warren is a demographer, sociologist, and as his website says, a dad. He's professor of sociology at the University of Minnesota, the director of the Minnesota Population Center, and he was the editor of the ASA journal Sociology of Education. Professor Warren has led several large-scale data collection and integration efforts, such as the High School and Beyond study, linking the 1940 US Census to modern aging surveys and the IPEMS CPS. Among other topics, his research has focused on educational gradients in mortality, occupational stratification across the life course, and panel conditioning in social science surveys. If you have not seen it already, I know that you will enjoy his 2017 paper in Annals of Epidemiology. Do inferences about mortality rates and disparities vary by sources of mortality information? Finally, before handing over to Professor Warren, I recommend to fellow graduate students to check out his website, which has many useful resources from his courses on population studies research practicum and getting your paper published. Thank you, Professor Warren, for coming to the Penn PSC.
Thank you for the plug for my website. And welcome, everybody. Thank you for coming. Let me get started here. I'm going to give a bit of an unusual talk today, at least for me. I'm not going to give you any results. We don't have any results for reasons that'll become clear in a moment. Instead, I'm going to talk about a cohort study in some detail. But along the way, I think it will raise a number of substantive and maybe theoretical questions about life course long processes and the way that social and biological factors interact, which I think should be of some more general interest. And I intend to leave as much time as I can for discussion because I think digging into some of those issues might be interesting. The work I'm going to talk about is highly collaborative. Here are several people who have been heavily involved. Chandra Muller and Eric Grodsky and I have been working on this for about a decade, with Jennifer Manley, Nicole Schmidt, and Ryan Demmer joining more recently. But I'll take the blame for anything that isn't right today. The premise of what we're studying these days begins with some empirical observations about disparities in dementia rates. This is women in their late 70s, but I could show similar graphs for women or men or for similar age groups. There are large disparities by race in rates of dementia, such that whites are much less likely to experience dementia in these ages. Similarly, there are large disparities by education in rates of dementia. These have been known for some time. What's not well known is why, and that's going to be a motivating theme of the talk I'm giving today. The fundamental question that we're trying to address in the field work that we're doing, and the data that will result from it, is how structural racism gets into people's bodies or across the life course and leads to dementia. A parallel question: how do early life disadvantages or traumas get into people's bodies and lead to dementia decades later? This is the model that is most commonly used to try to understand dementia or cognitive impairment late in life. Much of that research recognizes that education is an important predictor of cognitive impairment, but they think only in terms of educational attainment, which can be measured retrospectively by interviewing older people. There's also a recognition of racial differences in cognitive impairment, but there's a tendency to think of race as just a fixed and ascribed characteristic, much like genetic risks for cognitive impairment. This is sort of the public health model. And because of this model, and there's also focus on sort of genetic modifiers of the effects of educational attainment. But because of this model, here's where you intervene. You develop new drugs for dementia. You focus on late life therapies, things that you can do to keep your mind active, and the like. There haven't really been any effective new medical breakthroughs in preventing or reversing dementia for decades, but this continues to be in public health, at least, where people focus on about where to intervene to reduce these disparities. A social scientist or a demographer, which is what the leaders of this project are, come at this a little bit differently. First of all, we wouldn't think of race as an ascribed characteristic from birth. We would think in terms of structural racism. We wouldn't, and we would also then, going with that, we would think in terms of things like opportunity structures and social inequality and economic inequality and public policies that shape all of that, and childhood conditions. We wouldn't think in terms of race just as a category. This is the sociology of education, right? And that is what we bring to the study of cognitive impairment and dementia. That leads to a different set of thoughts about where we might intervene to reduce disparities in dementia. If we can intervene early in the life course around education policy or the social conditions that children face, perhaps then we can alter, in the long run, disparities in dementia. Of course, there's a highly interactive model here, right? Early life influences, social and economic and policy influences will interact with genetic predispositions. And so this is a complicated set of things to study. We also, in order to really understand what's going on here, have to think about the biology of these processes, about how structural racism or opportunity structures early on in life get under the skin, and how genes moderate all of that. A problem, though, in studying all of this across the life course it has to do with the nature of existing cohort studies. So in sociology, or particularly in education, most studies in economics, demography, we have great cohort studies, and they often start at birth or at least in school, and they follow young people through about midlife. I'm a sociologist of education. Some of the most important insights about education policy or the sociology of education have come from cohort studies that are amazing in their depth and end when people are in their 30s. And this is true for a variety of other studies as well. This is the kind of cohort study that we often have in sociology or economics or education. On the other hand, in the aging world, in the cognition world, they tend to have cohort studies that start middle adulthood. The health and retirement study, which is probably the most widely used study of aging, starts at age 50. You know about educational attainment because you ask people how far they went in school. You can get genetic risks because they have biomarkers, but we know nothing about early life before age 50, more or less nothing, except for a few retrospective questions about childhood conditions, but of unknown validity, frankly. So what we want is all of this. And this is where high school and beyond comes in. And this is what we're up to. So I'm going to talk about data and running a cohort study, but really it's these broader substantive issues and this somewhat crazy idea that early life factors and structural racism and opportunity structures are responsible for late life disparities in dementia. High school and beyond was initiated and initially led by James Coleman in the late 1970s with funding from the National Center for Education Statistics. It was a US Department of Education cohort study. It began in 1980 with nationally representative samples of high school sophomores and high school seniors. The high school and beyond cohort was followed up again every 2 years through 1986. It was a subset, about 27,000 randomly selected high school sophomores and seniors were followed every 2 years, including representative oversamples of racial and ethnic minorities, which will be important for our story. I said that the early life 1980s data are incredibly rich. There are repeated interviews with the students, of course, which covers every aspect of the kinds of things that are going on in their lives. There were interviews with parents, teachers, school district personnel. There are linkages to school transcripts at both the secondary and post-secondary level. We know a lot about these young people. The sophomore cohort was re-interviewed in 1992. The response rates to high school and beyond were remarkably high, even for the time. In school, it's easy to interview people, but even once they left school, response rates were still 85 to 90% through 1992. From the high school and beyond data through 1992, an amazing amount of very important sociology and sociology of education was done. Much of what we know about social capital or private school effects or curriculum tracking or high school students' employment and a whole variety of other things, at least initiated with high school and beyond data. The project High School and Beyond is now led by 3 sociologists of education, which makes sense given that it started as an education cohort. So that's high school and beyond through 1992, when people were about 28. It still doesn't tell us anything about late life or about cognitive impairment or the biology underlying any of this. The goal is to make high school and beyond cover all of it. And that's what we've been up to. So the first question when we decided to go back to the high school and beyond cohort is whether we could find people. We did about a decade ago, we initiated this, and of course it had already been 20 years since they'd been interviewed. Would they respond after 20 years? And who would pay for this crazy scheme? The who would pay for it ended up being the Sloan Foundation and the National Science Foundation and the US Department of Education's Institute for Education Sciences. And so in 2014 and 2015, we went back to the sophomore and senior cohorts, respectively, and we were able to locate people. It turns out there's no privacy anymore in the internet era. If you have people's names and birth dates and social security numbers and the names of their loved ones, you can find them almost all the time. But we were very happy to get about 2/3 of the cohorts to respond to a short phone and web survey in 2014, 2015, which was mostly about just updating their lives, school, work, health, family, sort of basic measures. Really, it was a proof of concept. Although we've done some important science with those data, it was really a proof of concept that we could recontact the cohort and carry it forward. So we've written a variety of papers, a lot of them on mortality, but also on educational continuation beyond the traditional ages of schooling and so forth. But that's not what the talk is about. So now we've got that part of the life course. Still nothing about late life cognitive impairment, still nothing about biology. So with support from the National Institute on Aging and the Alzheimer's Association, we are now starting on Thursday to recontact the cohort for a 2021 follow-up. The goals of the 2021 follow-up are to get the rest of this puzzle. So we need measures of cognitive impairment. The cohort is approaching age 60, too young for full-blown Alzheimer's disease, not too young for mild cognitive impairment, and certainly not too young for higher rates of cognitive impairment among people who, with less education, from racial and ethnic minority groups, and so forth. And the fundamental goal is to understand people's cognitive well-being today and to get a handle on these biological processes. So the team, led by a bunch of sociologists of education, is no longer adequate. In order to do these things right, and particularly to understand cognition and the biology underlying all of these processes, we had to diversify the team. And these are our current players representing a variety of disciplines, and there's their names. So how will we measure these things in red? How will we gather this information and supplement it, supplement what has been an education survey pretty much to this point? Well, let me spend a little bit of time talking about what the biological processes are that we think are most important to focus on. But then I also want to pay attention to how those biological processes are shaped by things on the left-hand side of my diagram. Education, not just attainment, but the processes of schooling and opportunity structures, structural racism in early life circumstances. How do those things shape the biology that ultimately leads to dementia? How are we going to measure those things? So let's start with genetic risk. It turns out that nobody, and I should say, by the way, I'm a sociologist of education. What I knew about the genetics or the biology of Alzheimer's disease 5 years ago, you could fit in a small car. I knew very little. So I'm going to present to you what I know, and if others know better or different, I definitely want to hear about it. So almost nobody is genetically predisposed to end up with dementia, whether it's Alzheimer's disease or some other dementia. Almost nobody is predisposed to automatically get it. Almost nobody is predisposed to not get it either. Environmental factors and genetics interact in really important ways to shape both the risk of dementia, but also resilience to genetic predispositions to dementia. And that's why I think this sort of cohort study has a contribution to make, because we understand, or we can see what those environmental factors were. So to get genetic information, sample members will spit into a tube, which has a preservative and a shelf. It's shelf stable. It can be mailed back to the University of Minnesota, where we'll do whole genome sequencing. So we'll get information about all 3 billion base pairs of DNA in humans' bodies, but we'll also do individual SNPs for things that are known to be particularly predictive of Alzheimer's disease. How will we gather information about the biological processes linking early life factors and later life impairment? I'm going to talk about 3 different categories of things. One is blood-based biomarkers of neurodegeneration. The other is the oral microbiome. And the third is COVID-19. So blood-based markers of neurodegeneration. If money and practicality were no object, if we had all the money in the world and everyone was fully cooperative with us, we would get everyone into a hospital and do a PET scan or an MRI, where you could see current biological markers of dementia here, which would look something like this. The folks on the right have clear markers of Alzheimer's disease. Of course, it costs, I think, thousands of dollars for each individual MRI, and getting people to universities or hospitals is much harder than getting them to respond to a survey. So that's not practical. It turns out, though, that at least in the last several years, the science behind understanding blood-based biomarkers has exploded. Pathologies that accumulate in the brain that are the definition of Alzheimer's disease or other dementias begin to accumulate years before anybody is symptomatic, before people actually show signs of dementia. And those pathologies can be detected in the blood, again, long before people are symptomatic. That's what we're looking for with blood. Blood-based biomarkers that indicate neurodegeneration even before people are symptomatic. There is evidence, although very limited because there isn't longitudinal data across the life course, that things like education influence vascular markers, inflammatory markers, amyloid beta and tau, which concentrate in the brain and cause all sorts of trouble, and biological aging. We're extending this to try to understand how all the things on the left-hand side affect dementia through these biological pathways. All of those biological pathways are detectable in blood via a variety of assays that can be done on the blood. So to do this, we are sending phlebotomists or health professionals to people's homes for a 1-hour home health visit. The phlebotomist will draw blood. They will also collect saliva for the DNA analysis if the person hasn't already donated saliva, and they'll do a variety of in-person physical exam type things. We're getting a lot of blood. It's about 4 tablespoons, which may not sound like a lot, but it's a lot. And what this allows us to do is to do the assays that are known to be predictive of Alzheimer's disease right away, but to store the vast majority of the blood. And the reason is that the science behind those assays is exploding. It's developing so quickly that we can't anticipate today what the best assay will be 3 years from now. And so we want to store most of the blood for future assays that have yet to be developed yet. Those are the assays on the bottom that we'll do right away that are the known biomarkers predictive of dementia, but we'll biobank the rest and take applications for future assays. While we're in the home with a home health visit, we'll get height and weight and hip and waist circumference, blood pressure, pulse, and a few other things. The second biological pathway is the oral microbiome, the creepy crawlies that live in everybody's mouths. It turns out that there are at least hypothesized links between the oral microbiome and any number of chronic diseases. And we're focusing, of course, in particular on dementia. How does the oral microbiome influence dementia, and how is it stratified by early life factors? I'll get into the how does it affect dementia in a minute, but it turns out that dental care and diet and where you live all influence the composition of the oral microbiome, the composition of the microbes that live in your mouth. And we know, or at least we hypothesize, that early life advantages and racism and education and opportunity structures and inequality and the like all influence access to dental care and diet and where people live. So it is reasonable to hypothesize that those early life factors influence the microbiome. This has only sort of been tested, and it's rarely been tested in anything like a large population-based study. The oral microbiome can influence dementia both directly through brain pathology. There are links between periodontal disease and dementia, which is a very direct connection in the brain. But there's also a variety of indirect pathways having to do with inflammation and inflammatory responses leading to neurodegeneration. Again, these things have been shown in clinics and laboratories, but never in a population-based study. Conveniently, the way to collect oral microbiome samples is with the same saliva kit that we're using to get human DNA. So we'll do microbial DNA extraction and sequencing on those same samples. And then the third biological pathway is COVID-19. I think we all know that rates of COVID infection and now vaccination are stratified by race and SES and education. There are a variety of hypothesized pathways from COVID-19 to dementia. Of course, we know relatively little with solid science about any of these, but these are all hypothesized. COVID-19 is known to lead to increased rates of stroke and brain inflammation, which can lead to dementia. There's some other pathways listed here. Many are biological, but I would also add stress and loneliness, which are predictors of dementia and certainly have been influenced by the pandemic. These speculations have led to speculation, again, that dementia rates may increase markedly in the coming years because of the COVID pandemic. So we're getting survey measures of who has had COVID, of testing and testing results, of vaccination, but we've also got information about economic impacts from reduced work and loneliness, and science knowledge for reasons I can go into later. But essentially, we want to understand whether people understand the science of viruses and infection, because we think that could play a role as well. And while we're getting blood, we'll also do antigen tests and return those results to sample members. So that gets us the biology of things. How are we measuring cognitive impairment? Well, what we're doing is a mixed telephone and web survey. People can respond either way, telephone or web, whichever is easiest. If you only had a telephone survey, you'd get very low response rates. We can't do clinical assessments. That would be the best way to get at cognition and to diagnose cognitive impairment or even dementia at this point, but that's not feasible for practical and financial reasons. And so we're going to rely on the telephone and the web. There are really well-validated ways to assess whether someone is cognitively impaired on the telephone. These methods have been used in the Health and Retirement Survey and other population-based surveys. In our case, we're going to do a variety of tasks. They sort of feel like games to sample members, including an immediate recall task. I'm going to read you 10 words. Okay, now tell me how many of those 10 words you can remember, like 1 minute later. A letter fluency task. I'm going to tell you a letter of the alphabet. In 1 minute, tell me all the words that you can think of that start with that letter. A category fluency task, which is an animal naming task. I'm going to give you a minute, name all the animals you can. A forward and backward digit span task. I'm going to give you a series of numbers, read those numbers back to me. Now I'm going to give you a series of numbers, read them to me backwards. And a delayed recall task. Remember those 10 words I gave you a few minutes ago? How many of them can you remember now? These feel like games, but they're all pretty well validated measures of memory and executive function. We're also asking a series of questions about memory complaints. It turns out people are often quite aware that their memory is getting worse and are troubled by it. So there are some good batteries of questions about memory complaints. Many people, I might even say most people, are not, however, going to respond to telephone surveys in 2021. Therefore, they will also have a web option. A problem, though, is that the kinds of cognitive tasks that I just named that are well validated on the telephone don't work on the internet, mainly because of cheating, right? If I give you a list of 10 words on a screen, you can write them down. You can sort of seeing them as a different task than just hearing them. So we need a web-based screener that is just a screener. Our goal on the web is only to identify people who are fine, who have no cognitive impairment. And so we will decide who is possibly impaired and who is certainly not impaired. If people are possibly impaired, if they're not impaired, they're done. We have our outcome. If they're possibly impaired, we invest a lot more money and time and resources to get them on the telephone anyway. We also send a random group of people who are not impaired to the telephone for methodological reasons. An important consideration, though, is that people are going to use their phones, they're going to use iPads, they're going to use desktops, they're going to use laptops, they're going to use old computers and new computers, they're going to use a variety of different operating systems. Some have good Wi-Fi, some don't. And so we needed to develop web-based screeners for cognitive impairment that would work in all of those environments. And so that's what we did. And so on the web, people do two tasks. One is a verbal paired associates tasks. I'm going to give you 2 words, officer, kettle. And I'm going to give you 24 pairs of words, actually. And then later on, I'm going to ask you, all right, which word went with officer? And if you've forgotten, it was kettle. So 24 of those, and then a visual version of that. Here's 2 pictures. Okay, now which went with the picture on the top left? 25 of those. And then the same series of self-reported memory complaints. In a separate cohort study, it's a cohort study, but smaller scale, more geographically, what am I trying to say? More geographically isolated, but for whom we know cognitive status, for whom we have clinical diagnoses of whether people are impaired or not impaired, we validated these screeners and they're highly predictive. It's very sensitive and specific measures. So we're confident in the ability of this cognitive screener to tell us who is not impaired. And that's really what we want. Not impaired, we don't need to invest any more money in them. This wave, anybody who might be impaired, we follow up with a telephone survey. So that gives us all of these measures across the life course, which I think is a first. There is no cohort study that has this rich early life information on a nationally representative sample and has these outcomes and measures of the biology underlying all of it. In addition to all of that, over the years, we've been pursuing a variety of administrative record linkages. So from back in the day, we had, as I said before, high school and college transcripts. Everyone's been linked on multiple occasions to the National Death Index, where we get both the date and the cause of death. I should say, interestingly, this is the AIDS cohort. It's the only nationally representative sample, I think, that has a large enough number of people who died of AIDS to do a population-based survey. We are seeking permission in the home health visit to link to pharmacy benefit manager records. It turns out that 80% of all pharmacies in the country report to just 5 pharmacy benefit managers, and we can get access to records from those, so we will know what prescriptions people have received over the years. We are a pilot program, one of a few cohort studies that has been allowed to link to state cancer registry data. So we'll know what types of tumors sample members have had and when. Experian credit reports. So we'll have things like credit scores and reports of bankruptcies. We've already linked to Catalyst voting records, CoreLogic real estate transaction records. These things are all amazing data. They're not particularly all relevant to studying Alzheimer's disease, but this is data that would be useful for a variety of purposes. We're pursuing linkages to Medicare and Medicaid records. They're too young for Medicare, but not for Medicaid. And this summer, we're pursuing a project to link the high school yearbooks, where not only can we see things about the structure of the school that we may not have observed before, but we can see things about social networks. And then by coding photographs, we can get indicators of things like attractiveness and skin tone and body mass index, as others have done in other cohort studies. So again, the substantive goal here, I've talked a lot about data and how we're going to collect data, but the substantive goal is to understand how education and structural racism and early life conditions of a variety of kinds of early life conditions affect the risk and resilience factors that shape dementia and that protect against genetic risks for dementia, and via what biological and socioeconomic pathways all of these things operate. And I would say a fundamental goal is to understand where racial and socioeconomic disparities in dementia come from across the life course, particularly as a function of early life opportunities and inequalities. And so that, stepping back a minute, the big picture question is whether there are places to intervene early in the life course to reduce rates of and disparities in dementia decades later, which may not be helpful for modern older people, but can help future generations of older people. So as I said, field work now in progress. We go into the field on Thursday. We did a pilot survey and home health visits with 200 folks just before the holidays. We're holding our breath as we go into the field on Thursday. Here are our funders, to whom we are very grateful. So with that, I will stop. And it looks like we have lots of time for question and discussion. I'm happy to talk about any of sort of the nuts and bolts because I'm knee deep in it. But I'd be happy to talk about the bigger substantive issues as well.
Thank you very much, Warren. This is really an incredible project, what you're starting. We have one question at the moment, and this is by Alain Haron. Alain? Alain? Okay, it looks like the speaker is, oh, okay, Alain, you need to unmute yourself. Okay.
Hi, good morning. I'm actually in LA. So I was wondering, what were the specific environmental factors that you're measuring? I was wondering if you're also assessing like self-perceived experiences of like discrimination or like racism, along with all of the amazing like genetic indicators that you're collecting.
Yeah, I'll answer those backwards. We do not have measures of self-perceived racism. It's the kind of thing we would love to put on the surveys that launch on Thursday. And it's the kind of thing that fell victim to those horrible meetings where you have to cut down all the ideas of the things you want to ask survey questions about because you can only ask so many questions. So we don't have measures of that. The early life measures, we have a little bit about everything. You asked what the environmental measures were, so I'm not sure if you mean literally environment, like exposure to lead or pollution, or if you mean more socioeconomic environment. If it's the latter, there's very little that we don't have measures of. If it's the former, we're a little bit more limited. We know where people's high schools were located, and that gives us some ability to link to administrative data on things like pollution sources or weather or things like that.
Yeah, thank you.
Thank you.
Thank you. Next question is by Hunjung Park.
Hello, do you hear me, Rob?
I do.
Yeah.
Good to hear from you, Hyunjun.
Yes. I mean, your work is always amazing to me. Like, you even think about this actually long span of life course actually to measure those things. I'm just just curious about, the sociology of education. We already know that, many things happen even before you enter the high school, right? At age 7, we already know that who are going to actually, pretty much have some idea about who is going to go to college and so on. So, I mean, if really wanted to spend whole life course, I mean, can we start from the birth course survey? I know that we don't have that, but
yeah,
in theory,
you're absolutely right. You're right. If we could pick our cohort, we would pick, one of the ECLS K or B cohorts, right?
Right.
We can't for a couple of reasons. One is we have access to the secondary longitudinal studies, not the birth cohort studies. The other is it's going to be, it was already an uphill battle. So I'm stepping back for a second. We're studying people who are in their late 50s. Most Alzheimer's researchers ask why, like they don't have Alzheimer's disease. Well, because you need to understand the precursors, right? You need to get baseline measures. You need to see cognitive impairment evolve over time. So it was already an uphill battle to start with a cohort that was only in their late 50s. To start with a cohort that's any younger, it would be very tough to get funding for it because people would argue, and I would say short-sightedly, there's nothing to study in terms of the outcome variables. I will also say, you and I as education researchers understand that there's a hell of a lot going on before people even start high school in terms of shaping inequalities that can be predicted later on. Back to that public health model, though, most of the people doing work on education and Alzheimer's disease have a hard time really conceptualizing why you need to know anything beyond educational attainment. Like, if I know they went to college, what do I care about middle school or curricular opportunities in high school or early pre-kindergarten intellectual opportunity? They have a really hard time conceptualizing why you need anything beyond attainment. So we've kind of opened that door and pushed back to high school. And I hope that this leads to kind of a field change so that a decade or 2 decades from now, people do go back to cohorts that we followed from birth, whether it's in the US or elsewhere, and really do that right. It may have to be in Sweden or somewhere where they have great administrative data. I don't know. Another problem, though, I'm sorry, I'm rambling now. We got lucky with high school and beyond, and it's because the National Center for Education Statistics didn't explicitly promise them that they would never come back to them. That's not true for NELS 88, for example, which is the next cohort in the sequence of education cohort studies. So I don't know if the birth cohort, if the ECLS birth cohort or kindergarten cohorts were promised that they were done forever or not. Probably not, but anyway, that would be another consideration.
I see. I mean, if I allowed it, I'd like to ask just simply the second question, which is, you measure mostly about the cognitive, the dementia, that part, but there is also the lots of adult literacy survey, right? In National Adult Literacy Survey, which actually show wide range of literacy skills, the adult literacy skills. So I'm just curious about, I understand that in order to understand the dementia, you kind of looking at the cognitive impairment, but not only just impairment, though, there's a lot of actually, the variations in the cognitive ability, which it seemed to me that could be another interesting outcome itself, especially, even if you have a college degree, there's still lots of actually the variation, which actually can lead to some outcome later in many years.
Absolutely right. Absolutely right. Yes. And we're already seeing that in the pilot work, that there is a hell of a lot of variation in the results of these cognitive tests, even among people who went to college or who had few early life disadvantages.
Okay, thank you.
Thank you. Jerry, your question was very similar to Hunjo's. Do you want to add something or?
Yes, Rob, very, very impressive work. And as Leanna said, my question is very similar, but I wonder, is it possible, for example, to link to birth records? Even having, birth weight, the kind of information that's on birth certificates, which, in a number of surveys in the US is possible. And
you are not the first person to raise this.
Oh, good.
And it is on at least the mental to-do list. It'll be challenging. So we do have obviously the person's own name from high school. So we have women's maiden names, and we have social security numbers and exact birth dates, which should be helpful. We don't know state of birth. We can infer that in most cases it was the state of high school, but it should, at least for some states that have more open birth certificate, records, it should be possible. Put it this way: if we can link to state cancer registries, I don't see why we couldn't also link to some states' birth.
No, it's challenging because it's longer ago.
It's longer ago.
Birth usually precedes the diagnosis of cancer. So this is a very useful nudge to keep pushing in that direction. I have an excuse to talk with Martha Bailey at Michigan soonish, and she, I think, knows more about birth certificate administrative linkages than most other people I know, and I'll be sure to raise that with her as well. Very good, but very impressive overall in any case.
Thank you. Next question is by Wendy Roth.
Hi, thanks so much for your talk. Really interesting project and very ambitious. I have a question about the biobank phase, and particularly about privacy and consent, because as I'm sure you know, there have been studies which have found that you can re-identify people based on their DNA. So it's impossible to make the data completely anonymized. I'm curious about how you mentioned that you don't know what kind of tests are going to be available in the future. And obviously biobanking is appropriate for that, but how are you explaining this to people in the consent so that they can give fully informed consent about their DNA being available and possibly used for any application in the future?
Yeah, we're very open. We're going to analyze your DNA to look for things that are predictive of cognitive impairment down the road, but we're going to store it, and it'll be available to researchers who will use it for scientific purposes through a secure facility. So we say what we can, which is access will be very restricted, and through NIA approved protocols and portals for accessing the information. But we're very clear about the risks, I think, and it's actually a separate consent process. One consent process is, can somebody come to your home and do these home health visits? At that time, you'll be asked to do a variety of things. You can say no to any of them. One of them, of course, is blood. But then a separate consent is, can we store it? And that one is, there are people who say no. You can come do the health visit, but no, you may not store it. And it's these kind of concerns. I guess I don't have a more reassuring answer. other than we provide everything quite openly and let people make decisions.
Yeah. And I know you haven't really started the data collection yet. I would have been curious about the rates of people saying no on the biobanking question, but I gather that hasn't happened yet.
Well, I mean, we did a pilot. Most people who, the real burden is the home health visit. Once people say yes to that, they 90% of the time agree to have the biological materials stored for future research. But we don't know what percent are saying no to the home health visit because they don't want, the risk of disclosure. I should say on the biological collection materials, we've unabashedly stolen all of the health and retirement survey strategies here, and we've actually collaborated with the teams of researchers who developed the HRS whole blood collection protocols, and we're using the same vendor and the same lab, and the leadership. So in a way, this has not been full scale piloted, but in a way, the HRS has done almost all of this. I also believe didn't have very high rates of people saying, yes, you can come for blood, but no, you can't store it. But again, we don't know if they're saying no to the visit because they don't want it stored.
Thank you. Next question is by Mary Yee. Mary?
Hello, yes, thank you. Thank you for a great presentation. I was wondering whether there might be any thinking about doing some qualitative research to accompany this fantastic collection of longitudinal data.
Not yet. Not yet. I will concede that we've been swamped with trying to figure out how to pull this off. I think that my gut reaction is that we don't know anything about impairment in this cohort, who is impaired, what their lives are like around that impairment, whether they have caretakers, whether they, and that this round is sort of a data gathering round, but that the next round of follow-up, which I would guess would be in 5 years or so, will be a little bit different. And it'll be different in a few ways. First of all, we'll have to bring some people into clinics and hospitals to do brain scans. But also we will know at that point who the people are who are in various boxes, if you will, people who appear to be cognitively impaired, people who have proxies responding for them because they're unable to respond, or people who are cognitively well, and maybe people who are cognitively well, but all the data would predict they shouldn't be. I could imagine identifying groups of people and then targeting them for qualitative, whether it's qualitative interviews or I'm not sure what exactly, but I think that might be a more appropriate place. We know so little right now. I will also say it'd have to be a different funding source, I think, unless it were focused more on caregiving. I think NIH wouldn't, but that's a practical consideration. That's a long-winded way of saying no, not yet.
Thank you. Next one is Snehamani.
I might have missed this, but would you be able to collect any information about, especially for the people you haven't reached, on the mortality status? And
yes.
If someone's dead, whether you know the cause of death, just to make sure you have a complete profile of the cohort being studied.
Yeah, the mortality work is first. You do that before you go into the field because you don't want to waste money trying to interview people who are deceased. So right now we are any minute expecting the results of linkages to the National Death Index. We did those in 2014. In fact, some of the first papers out of the project then were analyses of mortality. but yeah, that's going on now. Those data are fascinating for a variety of reasons. One is that for me, at least in a nerdy kind of demography way, there's not a lot of great research on the predictors of early mortality, early at that time before 50, now before 60, where you have prospective data with large samples, particularly a diverse sample. So I've been fascinated by some of the early work coming out on the predictors of suicide or homicide or accidents, which are the main killers of younger adults. And as I said, AIDS.
That's really interesting. Thank you.
Thank you. I have a question. So you said that you're using a lot of HRS information as a template, what you're doing. In terms of the cognitive measurements, are you trying to harmonize with the HCAP instruments? Because we know that HRS and sister studies, they're all trying to basically to use the HCAP instruments in all aging studies. So are you?
Yeah.
I didn't catch this exactly.
There are some of the phone batteries are parallel and quite intentionally so. The immediate and delayed recall measures. I believe they also do the category fluency and yeah. So several of the batteries are parallel. A difference is there are a couple differences. One is we're trying to be more sensitive to milder forms of cognitive impairment. So that has caused us to a little bit expand our batteries. The other is this mode difference. They have not done a full-scale web, and certainly not a wave where people could choose to respond by web or by phone, whatever was convenient for them. And so that has led us to have somewhat different measures. Mm-hmm. They're also bound by needing to be consistent over time, for good reason, across HRS waves. I don't know what they would do if they were starting over, but we have the luxury of starting from scratch. So yeah, there are harmonized measures.
Okay. So in
terms I have a question too.
Okay.
Sorry. Okay. No, very interesting. I have a question more about the process of moving from a study of inequality to thinking about Alzheimer and basically I want to see if, get a little bit more of the insider story about how that happens, and specifically I'm thinking that, and it connects with the discussion of mortality and other causes like suicide. How much of this is designed around one particular disease, like you're mentioning Alzheimer's, or is this, when you think about aging and old age, Alzheimer's is one of the multiple outcomes that you have. That is this really organized about just one disease, or is this going to provide more information?
No, it's a particularly strong study, I think, of cognitive outcomes, dementia. It's a revolutionary study of cancer also. And I think mortality in many ways. We have been primarily focused on cognition for a couple of reasons. One is that you need to get baseline measures, to study it over time. The other is that there's a lot of funding for research on Alzheimer's disease. And so that was a vehicle, and we're very appreciative of that support, and we want it to be everything it's cracked up to be for Alzheimer's disease research. But we've also been quite deliberate in making sure that it's generally useful for studying later life health. We have a variety of health. You kind of have to get a variety of health measures to properly diagnose cognitive impairment. People may score poorly because they're diabetic or they have mental illnesses, or you have to get a lot of health measures. But we've also been quite deliberate about making sure we're getting good measures of things like work and family and other kinds of outcomes. So we think of it as more of an omnibus survey that has particular strengths in certain areas, which also allows us to be flexible in the future. I hope that we can let it continue to evolve. And certainly the pursuit of all the administrative records we can get allows us, I hope, many more opportunities beyond cognition, right? So we have income measures, we have occupation measures, but with Experian credit reports, now we have really amazing, economic information that you couldn't get with a survey. So I think there are many opportunities beyond cognition, and hopefully more will emerge as we go forward with future rounds of data collection.
Okay, thanks.
In terms of how it got there, by the way, I am a little bit intellectually schizophrenic. People think I'm an education researcher, but a lot of times I'm actually an aging researcher. And it was probably Chandra Muller's idea to do this. Why are we frustrated with aging cohort studies that don't know anything about life before age 50? Why don't we go back and revive one of the education cohorts? It's probably her idea, but that schizophrenic in me allowed me to really grab onto that as an idea. There was probably beer involved initially, and it worked out. I mean, it's an exciting opportunity that I don't know. There was some luck involved, but it may be a particularly good example of why it's important to hang around with people who are not just in your area. I hung around with enough cognitive aging people to realize that their data had some limitations and that this was an opportunity.
Hey, thanks.
Great. We have one more question by Mary Yee. Mary?
Hi, can you hear me?
Yes.
Thank you. Yeah, so I was thinking, given the large demographic changes that have happened in the country over the last few decades, I'm wondering how you're going to account for this in your findings, especially taking into account increased racial ethnic diversity and the life experiences of immigrant groups. And I'm thinking especially non-native speakers of English.
Yeah, well, we are confined to this cohort. So we can represent the experiences of just this cohort. This cohort is diverse, but in different ways than modern cohorts. There are a fair, I don't know the number offhand, but a pretty substantial number of immigrants in the cohort. As I said, there were random oversamples of African Americans, but also of some Latinx groups, including Cubans, which given the situation in the late 1970s isn't so surprising. But the truth is that we can't represent modern birth cohorts. We can't talk about diversity as it is for younger folks today. We can only confine ourselves to the diversity in that generation. And so it's a matter of being very clear in our findings that there are things that we can say that people haven't been able to say before, but there's a lot of things that we won't be able to say.
Thank you.
Thank you. So this is such an incredible cohort, such an incredible work. And before we close, I was just wondering if you can tell us how can one get access to the data?
Sure.
What are your plans for sharing data?
Yep.
When will be
Sure.
the data available, and etc.?
I would guess we're probably 2.5 years from data, or maybe 3. The survey data will be accessible still through the National Center for Education Statistics restricted data system, which is how they've disseminated all survey data since 1992. The genomic and biological information will be available through the National Institute on Aging's I can't remember what the acronym stands for. NIA has a preferred vendor for making available and harmonizing genomic and also other biological material. The administrative data we're still working on, and it often varies by the source of the administrative data. So state cancer registry folks are very particular about how their data is distributed. CMS is so we're still working on the administrative data. It'll be complicated because you'll have to work in restricted environments. Looking ahead, a next step on our end is to build project infrastructure to facilitate access, to make it easier to work with people so that I'm not the one answering all the emails. There's somebody who can really dedicate themselves to to facilitating access for users. Thank you all for these really interesting questions and for being pushed in certain directions.
And thank you so much for your presentation. So this is really incredible work. It was very, very interesting to hear what you're working on and what you plan to do. Yeah.
Well, thank you for inviting me.
Thank you, Rob.
Thank you.